before was the more popular one, the next-gen geospatial. But welcome. I am Michele Cosi, and also welcome to every person that is online. And it says next gen right here, but it should say bioinformatics and genomics now. So we're switching things around. I wanted to start by, first of all, thanking you for being here and introducing myself. My name is Michele Cosi, and I am a Data Science Institute instructor for, well, the Bioinformatics and Genomics series. I'm also a maintainer for a lot of the tools that we're going to be seeing throughout this web series, or if you're in person, through this in-person workshop series. What today is going to be about, I'm going to show you some of the tools that, not the tools per se, but the machines that we're going to be using throughout our time here. And these can be broken down into two: the HPC and an online platform called CyVerse. If you did read some of the prerequisites, I'm asking you to know a little bit of genetics and genomics, and I'm going to go around the room soon to figure out what your backgrounds are, and to also know a little bit of command line if you know any of that. So, hi, may I ask you to introduce yourself or give a little bit on who you are?
Hi, Haley. I'm a master's student in department, also department. And I feel pretty good about genetics and genomics, but don't really know command line. But hopefully, like end of this semester, maybe beginning of next semester, I'll have some like IEC data to work with.
Sure. Cool. Fantastic.
My name is Sarah. I'm also in the EDB department. I haven't worked with genomics data before, but I'm going to soon.
Okay.
So I'm familiar with like at a broad level of like a lot of the concepts and pipelines and stuff, but never actually, done hands-on.
Fantastic. Then this is exactly for you. This is what we were aiming for. Some graduate students with a lot of, or with some biology background, trying to dip their feet into genetics and genomics, bioinformatics. What I'm going to do throughout the next few weeks is introduce some tools for transcriptomics and RNA analysis. You might have heard of StringTie, you might have heard of the Sec2. We're trying to go through those. But before we do get there, I need to show you what we need to use first. And that brings us to today. One of the things that I would appreciate going forward is if you do have a laptop, take it with you, simply because it's easier to code along together. And also, if you have issues, for example, logging into the HPC or understanding how the HPC works, or CyVerse, this is something that can really help. Having a laptop can really help. Jeff has shared a link to the Zoom, and I'm supposedly thinking you guys you're not on the Zoom right now?
Yeah.
Okay, so if you could, it would be a great way for you to gain some of the information that I'm going to be sharing. Yep, thank you. Also, for the people online, if you do have a question, you can go ahead, unmute, and ask the question, or just put it in the chat. Thank you. You sent this one to me?
And everyone can on this Zoom, right? Just go to the
Haley's here.
Is it shared on GitHub?
The Zoom link?
The Zoom link, I can.
Or I just
Yeah.
okay.
Yeah, it's in the email. So I'm gonna give a minute for you guys to get on. Okay. Whilst you do get on, I'm just going to explain a little bit what's going on over here. So most, if not all, of our content is shared and open source on GitHub. Familiar with GitHub? Yes, no? Some little bit? Yes, right. This is where a lot of our content goes. And in this specific page that I'm sharing right now is the description for every single lecture, every single, I can't really call it a class, but every single workshop that we're going to be hosting over the next few weeks. Each of these also comes with a link to a more in-depth resource, and this contains everything that we're going to cover through each day. I'm going to be updating the link as time goes by, and also every single lecture is recorded and posted on YouTube. So if you can't make it into one or not interested in one, but want to still hear from it, you can always go and look at it in your own time at home. So I believe that does cover the extreme basics. So where all of the information is, all of the materials, and essentially the overall schedule. So it starts with platforms and then tools and then pipeline management. That is something that I like myself because pipeline management can truly help you with whatever you are working with. I'm a fan of Nextflow. Some people like SnakeMake, and therefore we're going to keep those two one after the other. DSEC2 is something that we also are going to be covering. I'm going to be handing it off to my colleague Carlos. And we're also going to be talking about how to draw pretty graphs using, well, R. We're going to be talking about current trends in bioinformatics, so AI and ML informatics, and using MLflow later on in the series. Now, stepping back and walking to today, talking about today. The last thing, what I have set up here is a visual representation of what I'm going to be doing. Every snippet of code I'm going to be reproducing, and you're welcome to reproduce it as well. I'm zooming up and down really quickly right now because this page is what I will be using to base my today's lecture on. But at the same time, I'm also going to be calling up on my terminal. So if you do want, you can code along as well. Now, cool, it's 3:10. And the next thing that we're going to do is that we're going to talk about the HPC. Don't worry about the times. I got the times wrong. And right after we talk about the HPC, we're going to be talking about this other little platform called CyVerse, and I'm going to be very snappy about it because we're going to be using that a lot more in the few coming weeks, and you are going to get very familiar with it. Okay, seems like the both of you here are using a Mac. Are you using a Mac? Is that a Mac? Okay, no problem. You should still have access to what we call a terminal. PowerShell is a form of terminal, and but you are not forced in any shape, way, or form to follow along. Why do we want the terminal? Well, very simply, a lot of these tools that are for bioinformatics are solely based on command line and command line tool. From my end, this is what happens when you have biologists do the computing and computer scientists do the biology. Everything just kind of go and nothing really works. So if you do not have a terminal, that's completely fine. But for next semester, no, sorry, for next time, I do need you to create a CyVerse account, which was also one of the requirements. If you had time to play around with it, great, fantastic. If not, you'll have time to do it later today. The outcomes for today are to understand the structure of the HPC and very simply log in, understand what a bash script is, understand what Slurm is, and maybe we'll even be able to ask for some, what do you call it, some compute. Then I will talk about CyVerse, but also something else called Jetstream if we do have enough time. I know that some people online are not from the University of Arizona. It is extremely likely that your own system is based on something similar that the University of Arizona runs. So even if you can't follow along, even if there is some changes between the various paths that you guys have and us, it's kind of the same. So try to feel your way around it. Okay. Before jumping into the HPC, bioinformatics and the need of compute. The field of bioinformatics has become increasingly reliant on computational power. Can you tell me why? Too much data. That works. Exactly. 2003 to, I think it was 2003 to 2006, the Human Genome Project took years and millions of dollars. Now you can walk into 23andMe and get your entire DNA scanned for $100. So things have exploded exponentially. So too much data is exactly the right answer. Too much data and the complexity of the data also has improved, which has led us to understand that we need proper platforms. One of the platforms that we are going to be using is excuse me, I have to admit somebody in the chat, is the HPC. We have one here at the University of Arizona. HPC stands for, yes, high performance computing platform, but that doesn't really matter. But nowadays we live in 2024, and there also is an alternative to that, and that is cloud computing. Not everything needs to be talking to each other using what we call a Slurm scheduler or a PBS scheduler. We can also do things with something that's called Kubernetes. I'm spitting out jargon, but I hope you'll understand later what I'm talking about. Here at the University of Arizona, we have an extremely powerful HPC platform. And I'm going to give you a 10,000 feet high view of what the HPC looks like. So what do you imagine the HPC looking like? If you have ever seen one, or if you can imagine one. That's also open to you, by the way. Not you, okay.
Like a Borg cube.
Like a Borg cube, okay. Interesting. Maybe probably not far off, since it's literally a room full of computer number 1, computer number 2, computer number 3, extremely boring stuff. Bioinformatics is boring, everybody. But I don't know if you went off of what's here on the screen, but it's kind of like that, you know, it's a cube that has CPUs and RAM in it. But the thing about the HPC is that you can't just connect to it. There is steps that are required for you to connect to it. And I'm gonna get into the weeds in just a second. The first thing that you do have to understand is that in order for you to connect to the HPC, you will need to do so through something called SSH. SSH stands for Secure Shell, and it's a protocol that allows you to connect to a remote computer. So you're University of Arizona students, I assume. All of you don't know this, but all of you have the ability to connect to the U of A HPC. How do you do that? Well, let's go to a little terminal here on the right. I'm going to make it a little bit bigger, so perhaps it's easier to see. I'm also going to move these around. Thank you, Slack. Disappear, please. Thank you. We're gonna call on secure shell. To do that, you do SSH, and then you add your University of Arizona username. So for me, it's Kazi. And then here is where the magic happens, hpc.arizona.edu. Notice how this is very similar to just a URL. And this is literally the address for the HPC itself. Typing in SSH hpc.arizona.edu and pressing enter will prompt the second password. It's asking me for my password. This is University of Arizona password. I'm just going to press enter here. And now it's going to ask for my dual factor login. Alrighty, no problem. I'm going to reach over to my phone. When you do get to this point, you want to press 1, where on your phone, you're going to get a little notification where you do approve people. Now, I have perhaps gotten myself a little too far. When, for example, you were typing in your password, if you were following along, nothing was coming up. Why? Well, simply speaking, security. When you do type in your password on a Linux or a terminal, sorry, it doesn't show up. So the characters are also hidden and whatnot. But before I do go further, I want to ask how we doing? Even online, if you're following along. If I do hear crickets, that's fine with me.
Yeah, I managed to log in.
All right, fantastic.
They're not able to log because the PI is not sponsored.
That happens after. Yes, after the login. Without the PI, you can ask for resources. So that's something I'm saying. Oh shit. Oh. Okay, we have one person that was able to log in. And I need to hide you. Go away. So what is going on and where are we? What you're seeing on screen right now here on the right is not actually the HPC just yet. This is something that we call the Bastion host. And what is a Bastion host? Well, I'm going to pull up this little infographic right here that I have gotten from the University of Arizona's HPC website. We are here now, the Bastion host, and the Bastion is exactly what it sounds like. It's a place where it stops people from logging in, such as back in those days when there were castles and whatnot, you will have a bastion, which will stop attacks from entering the castle. Imagine the internet as this one place where there is humans, but the humans are only like 1% of it, and the rest of the internet is like porn sites and bots. All of these things are trying to get inside the HPC. They're not thinking, they're just attacking it. And the Bastion host is exactly what keeps them from entering. So once you are in, it does say, hey, type shell to access the job submission hosts for all environments. Okay, computer, I'm going to type in shell and press enter. Oh, what is this? Alrighty. The computer now understands, oh, you are human and you want to access to the rest of the HPC. Here are your options. You can either go to Puma, Ocelote, and El Gato. But what are Puma, Ocelote, and El Gato? These are the free clusters that are part of the University of Arizona's HPC. It's going to scroll down a little bit. The newest one is Puma, and it's been around since 2020. I've had the pleasure to test it out, and it's honestly, back in those days, it was the fastest thing on the planet. And I recall those days in good faith. Ocelote was purchased in 2016, and it has many processors, but is still slower than Puma. Makes sense. And the oldest one around is El Gato, which has been around since 2013. He's 11 now. Time to start going to middle school, I guess. And the reason why we do have three is because there is three different types of compute that you may need. Puma, being the newest one, is also the strongest. Elgato, no one really does use Elgato, which is exactly what we want. We want a system that nobody uses so that we can go and learn things. So if you are in this specific, if you are following along and you are in this specific step, you are going to type in Elgato. You're going to press enter. And as you can see on the left over here, it switched from Puma to Elgato, and that is a confirmation that we are now in the Elgato system. Believe it or not, we're not yet still using the HPC. We are just getting through the motions to get to the HPC. I'm going to scroll up a second because I wanted to show you where we actually are. We are here. We are at the login node. And just to get here, we've had to go through SSH into the Bastion host, choose a system through shell, and we are the login node, and the login node presents itself as, well, nothing. It's just an empty shell. But what can you do in the login node? The login node is the place where you can do two things. One, launch jobs, or two, ask for an interactive node where you can do your jobs. One of the most popular tools in bioinformatics is something called BLAST. Raise your hand if you heard of BLAST. Fantastic. What does BLAST do? Yeah, it's a sequence aligner. Excuse me?
Just it finds matching sequences.
Finds matching sequences.
Unknowns, yeah. Sometimes unknowns, sometimes knowns. Sequence data.
Mostly unknowns, honestly. And how do you launch BLAST? What is the command for it?
Blast.
No. Close, though. Trick question, because there is two ways that you can call for BLAST. You can ask for BLAST N, which is the BLAST for nucleotide sequences, or BLAST P, which is for BLAST protein sequences. So if you type in BLAST N in this specific case and press enter, Elgato tells me, command not found. I don't know. I can't find it. But this is the HPC. This is where everybody should be able to do all of their compute. Well, this is the login node to the HPC. You're not going to be able to actually get to the HPC. You haven't actually gotten to the HPC, and you haven't actually been able to use BLAST just yet. The next step is then being able to get to the HPC itself. But before we do that, it's very important that we talk about money, because money makes the world go round and it's really annoying. In this specific case, money comes in the form of allocations. Allocations on the HPC can be viewed using the command VA, view allocations. So if I type in VA here, it's going to give me a long list of a long list of PIs that I belong to, and tell me how many allocations I have every month. These allocations reset every month, and I want you to look at the last one here specifically, because what does this tell me? Well, this tells me that this month I have 7,000 hours worth of compute to go. What does that mean? That means that if I'm in a lab with 5 other people and everybody's using the HPC, everybody's going to talk to each other, because those 7,000 hours are going to run out extremely quickly. It looks like it's a lot, but 7,000 hours can be so and so. It tells me that I am part of a group called Data Lab, and it's extremely important for you to know what group you're in, because essentially that is going to be the group which you are going to be, quote unquote, billing when you're launching an analysis. Excuse me. Tells you also for how long it's run for. But these are the two things that I did want you to know, that HPC does run on money, and it needs to know who to bill at the end of the day. Make this small again. We don't necessarily need to talk about storage right now, but the HPC is a good place to store things for the short term. Your home only has 50 gigabytes of space. Your group, so we talked about group over here, has roughly 500 gigabytes of space. But if you are part of a larger research, your PI needs to send in a request, and this is going to give you an X disk allocation, which is roughly 20 terabytes, and you can ask for more if you really need it. As bioinformaticians, I don't really think we're gonna go past 20 terabytes. But things are funny, and if you do work with image data for phenomics, then you're definitely going to pass 20 terabytes. Now, before we launch our first job, I'm going to expand over here because we also need to know that there is other people in here. And how do I check what other people are doing? I type in a really important command called sq. Q shows you the queue, and YS, well, that stands for Slurm queue. Slurm is the scheduler that runs on the HPC here. And I'm gonna press enter. And I can see that there is a lot of people right now on here, but not that many. I can see the user Catherine using an RStudio, and the RStudio is running, and it's been running for 1 hour and 25 minutes. Believe it or not, you can have enemies on here, and it's insane because like you want to use the HPC, but then you see Amiroyue always taking all of the GPUs and doing all of the analysis, and you never get to a running status. You're always on waiting and the queue. And so you log in and then you see Attila and it's like, you're running for 1 day and 8 hours. No way. What are you doing? So you actually go on the list of staff and actually try to find it. So you make enemies through SQ. It's dangerous. Be careful. So that's how you check how many people there are or who's running, how busy the queue is. SQ. I'm gonna scroll down a little bit because now that we know how to bill and the allocations available and how to check if there is any space for you to run things, we can start looking at how to launch a specific analysis. And in the interest of time, I'm not going to go too far in here. We do only have an hour each week, so I'm just going to look here on the left how to actually launch a specific analysis. So there's 2 ways for you to do your analysis. One is called a job submission through a batch. So for every job submission, you need to write a small little script. Have you ever done any scripting whatsoever in your life? Cool. How do you start scripting? What is the first line you write when you do a little bit of scripting? Pound, shebang, /bin /bash. Yeah, cool, fantastic. That's exactly what this sort of scripting is. It's a quote unquote shell script. I'm going to make it a little bit larger. Oh, gonna make it a little bit larger so you can see what's going on. It's a quote unquote shell script, and you can literally have any extensions that you want. It can be .sh, it can be .slurm, it can be .old, and it won't affect it. But what is important is that after you call on that first line, shebang bin bash, you add the following S batch lines. And again, S batch in this specific case stand for slurm batch. And the absolute minimum requirements are that you give it a name. So job name, in this specific case, blast job. You give it a partition. Partition is HPC lingo for, am I going to go, do I really need these resources now, or can I wait? Standard means that you kind of get them soon. High priority means that you need them now. Obviously, those are more expensive. And windfall means that you can get them a lot later in the day. I have classes now. I can check this later. 3, you need to indicate how many nodes you require. Nodes, and there is a different amount of nodes per HPC. So the Puma HPC has like 42 nodes. Elgato has 16, so please be kind. Here, and you know, you can just use one. Then you have to identify how many tasks per node. Yes.
So is the number of nodes you would use just based on like the computing power you need, or why would you use more than one node?
Yes, it's based on the computing power that you need. Yes. Again, I don't want to get too deep into this, but it does get quite mathematical. For example, let's say that you want to assemble a genome. If you want to assemble it, rice. If you want to assemble a rice genome, 2.92 gigabytes, and running it on one node, like, dude, it's not gonna go anywhere. Running on 16 though, that's a different story. And then you're running on 16, and then you want to figure out how many tasks you want per node. You can do some maths to really maximize things around. And again, CPU per task. So one node can have, let's say, a 16 cores maximum, let's say. Okay, I don't have the list in front of me. But if you're able to do, like, for example, 3 nodes and 10 CPUs each, things are going to go run a lot quicker. The computer are going to talk to each other. Regardless, these are the 6 different lines that you require when writing a batch script onto the HPC. Job name, partition, node, task, CPU per node, amount of memory, and the total time that you want to run in. After that, you want to run something called modules. Modules are what is necessary for you to call on specific tools. Earlier, I showed you that BLAST did not exist. Well, that is because I didn't load it. You cannot load modules onto the login node. You can only load modules within your jobs. And right after that, you need to start scripting for the rest of your analysis. This right here is literally a really simple BLAST job. In one of my other workshops, when we have more time, I actually run it, and it's fun. You get a little BLAST result with it. You then submit it using sbatch. And what you will then see is that it goes into the queue, and you will see your name right here at the bottom, and you're going to reload it multiple times to see whether it actually is running or not. The other way that you can launch a specific job is through something called the interactive node. And the interactive node gives you access to a command line, essentially, and one of these nodes in one of these nodes specifically. Very similar to the as batch, you have to ask for the interactive node. For example, how many CPUs you want this interactive node, how many gigabytes per CPU. And this is a command that you submit to the command line. Or if you just want a quick and dirty, you can just type in interactive and press enter. And this is going to give you a small little allocation. I'm going to expand this for now. It's going to give you a small little allocation in a quote unquote free account. It's the windfall account. And because you are on Elgato, is actually quite quick to actually get to that. If you do interactive on Puma, it takes forever. So as you can see, it runs for 1 hour and it only has a single task per node. And it only has one node with 4 gigabytes. This is literally a very small computer, but this is a good example for showing you how modules work. So again, if I were to go here and type blast n, it's not going to find it. But if I were to do module list or module, let's do module list first. If I type in module list, it shows me the modules that currently are loaded. I can go ahead and do module spider, and kind of like a web shows you, hopefully, please don't break. Oof, there we go. It shows you a list of all of the modules that you can load. So I just said something interesting there. I just said load. Oh, so I type in module load blast, press enter, and I see that it didn't complain. And when you are running on the HPC and nothing complains, something happens. So I'm going to type in blast n and see what it tells me. And hey, blast query options error. Either a blast database or subject sequence must be specified. So what happened? I'm going to take a step back a second and go back to this specific image right here. What happened? Within the last roughly 30 minutes, we got our laptops out and we used SSH in order to get to the Bastion host of the HPC. From the HPC Bastion host, we then typed in shell, which showed us a number of login node possibilities and options, excuse me. And we decided to go for one of them. We decided to go for Elgato. Within Elgato, we were able to look at the available allocations. We were able to look at our specific groups, if we have any. Beg your professor to add you to your group so that you can actually use the HPC. And from there, we said, hey, I want one of these small computers. Can I try it? The computer said, the HPC said yes, and it gave us this one node right here, just a random one. And from there, we were able to say, hey, I want to do some blast. I know I checked your packages and I see that you have blast there when I did module spider. So by doing module load, you now loaded the tool blast. This is 10,000 feet view of what the HPC looks like. But honestly, you work in geosciences, you work as a geneticist, you work as a physicist. It's all like that. You go through the same pipeline. And this is something that I wish I knew when I first started my bioinformatics work, how the HPC works. I was really scared to use it, but through time, I made enemies, and I'm happy with that. I'm gonna take a second and check in with everybody here. We doing all right? Okay. Any immediate question, burning question regarding the HPC?
I have a quick question.
Yes.
In my terminal, it says that there was a client loop send disconnect broken pipe. I'm wondering what that means. Because it wouldn't let me do the interactive thing.
I believe you have an internet problem.
Okay, could be.
Yeah. Try again, honestly.
Okay,
Yep,
thanks.
no problem. Client loop is usually an internet problem.
Yeah, SSH.
Yeah, yeah. Okay, so the HPC is an amazing resource that we do have on campus, and it's what we have right here. When is the best time to use the HPC to reduce enemies? That's a really good question. Don't use the HPC during exam time. Just don't. That everybody has their exams, and they launch so many jobs to trying to figure out and finally get a C out of it. Not during late December, never use the HPC. During the day, launch things early in the morning. And I mean early in the morning, I mean 8 a.m., maybe 9 a.m. Late at night, perhaps, but late at night, I mean 9 p.m., 10 p.m., because anywhere between 3 p.m. to like 6 p.m., it's busy. So busy. Everybody's just trying to launch things. So hope that helps. But okay, the HPC is a fantastic tool that we have on campus for you to use, and perhaps you will have a chance to use it when you actually do need it. And I'm going to show you another platform today. And this is going to be the platform that I am going to be using to show you all the tools throughout. Welcome to CyVerse. I'm going to now deviate a little bit from the little thing on the left. This is where I have all of my notes. But I'm just going to talk through it. So I'm just going to make this bigger. If you do have a CyVerse account, please do log in. If you do not have a CyVerse account, go to user.cyverse.org. Actually, I'm going to open up an incognito and go to user.cyverse.org. I'm going to copy and paste this and put it into the chat, and you can go ahead and sign up and log in there. Please do use your EDU account. This is going to save everybody money. Thank you very much, and give us good metrics. Where are you, chat? If you do not have a CyVerse account, go ahead and type in user.cyverse.org and create an account. If you do have a CyVerse account, go type in de.cyverse.org, which is going to get you to the discovery environment. And now my extremely quick spiel on CyVerse. CyVerse was born as the iPlan collaborative back in 2007. And for the next 15 years, it grew out of its plant biology core and renamed CyVerse. Why? Because people from, for example, the space sciences looked at us and said, hey, we want to use CyVerse, but I don't like that it's called iPlant. So, well, the name was changed in 2016, and we gave it a nice little fresh coat of paint in 2019. And as of 2024, we have, I think, 4 external CyVerses, where there is one in Austria, one in the UK, one in Canada, and one in Australia. CyVerse runs on something called Kubernetes, which is containers and, well, let's just call it cloud computing for now. I am digressing, but what I want to show you through CyVerse is what it can do, other than throwing errors. CyVerse is a great place for you to deposit data. As you can see right over in the home, I have deposited roughly 2.8 terabytes, and I have 5 terabytes. Every single person that is affiliated with the University of Arizona does get a quote unquote pro subscription to it, which is roughly 3 terabytes of space. And that is a great place for you to put your data. It also gives you, very similar to the HPC, CPU tokens. And CPU tokens, well, that means that you can run a specific analysis for a specific amount of time, and hopefully nothing breaks and nothing fails so that you can run more. Now, I do want to address that there is a big difference between the HPC and CyVerse, where if you want to use the HPC, you require a project that your professor is working on, whilst a CyVerse, you don't need permission from your PI to use it. So you can actually go ahead and try yourself. Now, a quick tour here on the left. I have an expandable menu where I can go to home, where I am now, where I go to data, where I have all of my data, share data, data that's shared with me, and data that I can share with my folks. Apps, and this is where I launch applications that I can then use for analysis later on. Analysis, which are running applications that I can go back to. A Cloud Shell. And Cloud Shell is extremely important because it allows you to step away from using the mouse and use the keyboard. And when you do use the keyboard, all of a sudden you have more flexibility. And Teams is a way for you to share certain data with a specified group, given permissions and such. I don't think we're going to be covering Teams for today. Today, I want to show you two things. The first being data, and the second being apps. I'm gonna go and click on data. Well, it's pretty straightforward. I think you may not have as much data as I have, but here on the left, I can see my own folder, or folders that have been shared with me, or folders that are available for everybody on the internet.
Yeah, you stop sharing?
Excuse me?
Oh, never mind.
You're fine.
I'm not seeing what you're seeing.
Okay.
Don't worry about it. Sorry.
All right. There is different ways to upload your data, and they get more complicated the more data you have. The less data you have, you can use the little upload button right here, and it's a fantastic way to just get data to you. Just do know that this little upload button is limited by, well, the power of your browser, which isn't so much. An advanced way to transfer data through to CyVerse is by using third-party tools like CyberDuck, cyberduck.io/download, and it's cute. It looks like a little duck, but what it allows you to do, I have to move Zoom, is that it allows you to connect to the data store and view the entirety of your files from your computer, like a literally folder. And you can drag and drop files from your computer to your online repository of on CyVerse. And lastly, it's a tool that I'm going to just talk about. It's called Go Commands, and it's the best way to transfer data to CyVerse. It's quick and it's snappy, but it is a command line tool, which means that you do have to have a little bit of command line knowledge. You can upload data, you can download data, and you can access your data through the apps. I am now going to go to the apps folder right here, and you can launch any of the apps that are available here. And these range from an Ubuntu desktop to an RStudio to different types of Jupyters. Have you ever used Jupyter? Cool, then I am going to hold my horses a second. Jupyter is a little bit like an IDE. So it allows you to access, it allows you to share data, to share code with your coworkers. And it also allows you to, for example, share notes with your coworkers. Very similar to a shell script, but for Python, for RStudio. And I'm going to launch one right now so that we can look at this better. I suggest you do launch the Jupyter Lab Data Science if you want to. It's the one that I use for demos. But for next week, I'm going to be sharing one that I have made with you, which is the Biosciences Jupyter Lab. This is what a launch window looks like. I can give my analysis a name. So I'm going to give it today's date 0903. I can add comments if I need, and I'm going to leave the output folder as is right now. The next page is very important because I can choose what the minimum and maximum resources I can use. I can select up to 64 cores or a minimum of 1. If I do select a minimum of 1, the system is gonna go with a single core. It's not gonna pick 64. But if I do want 64, then I can select the minimum being 64 and the maximum being 64. Right now, I'm just gonna go for 4. Memory ranges from 2 GB to 128. I'm just gonna go for 16 right now. And I'm just gonna click next. I don't need any disk space right now. Over here, I am now going to click Launch Analysis. This is just a summary of the tool before it launches. And now it's gotten on something that's very similar to the queue. And we're going to go see it. We're going to click on Go to Analysis. And it's already past the queue. And now I'm here in my Biosciences online platform on CyVerse. I have access to a terminal. I have access to a notebook. I'm going to show you what a notebook looks like. And I have access to RStudio. I'm assuming you all know RStudio. Okay, fantastic. So I'm not going to open up RStudio, but I'm going to go and fetch. Actually, I should have some. Show me. Yes, I'm gonna open up this Gradio workshop just to show you what the Jupyter Notebook looks like. So Jupyter Notebook is literally a notebook. And in your notebook, you add notes. You add small little drawings for what that may help you in the future. A Jupyter Notebook allows you to very much do the same. So, for example, type in what you want to run. In this specific case, pip install Gradio. Oh my God, there's a lot. But also add in blocks what you want to, what your code is. Import Gradio, define a specific function, and run a function. You could also add notes as it is over here. And the thing is that you can save this and give it to your coworker so that this person can run things over again. So Jupyter Notebooks are a great way to share code and make your work reproducible. I'm gonna close this one right here, discard, and open up the terminal. Very similar to what we saw on the HPC. This is a terminal where, well, you can launch Blast. Yay, same issue as last time. No problem. That's exactly what we're looking for. You can launch Bowtie. Oh my God, what are all of these? This is something called flags. What I will try to cover is for specific tools, I'm going to point out which flags you should worry about, what scores you want to think and keep in mind what the thread is, if that is something you are interested in. So I'm going to be honest, for next week, I did put in massive list of tools that I will be covering. I'm not going to be covering all of those. I'm just going to cover a subset of them because those are the ones that I think will be more important. What your job is to tell me, Michele, I don't care about BLAST. I want to know about string tie. So I will put in string tie and tell you about it. So it's 3:57, and before we go, I need to share this with you so that you can tell me what you want to see. And I need to share. No system view. Cancel. No system view. Like, catching that. I'm going to copy this, and I copy this link right here so that I can put it on Slack. And I want you to please fill this in, not entirely, you don't have to do all of it, to tell me, hey, I care about this, I don't really care about that. You kind of suck today, but that's all right. So please, I'm going to stop my share and thank you all for your attention today. The goal for today was to show you some of the resources that we have on campus to do bioinformatics, and these are, well, the HPC and CyVerse. We will be using more of CyVerse as we go, and simply because I know things work there. And by all means, do fill in this form and tell me what you want to hear. Tell me what tools you want me to cover. Tell me what your quote unquote major is and what we need to focus on. If we are focusing more on transcriptomics or focusing more on genome assembly. The tools that are used for both are slightly different, but the pattern is the same. So thank you very much for your attention, everybody. I hope to see you next week. And by all means, have a great week.
Thank you.
Bye.